Welcome back to the Sand Science Experience, where we explore the most exciting new research papers and preprints just for you. Before we dive into today's really mind-bending topic, please take a moment to like this video and subscribe to the channel. Your support truly helps the Sand Science Experience continue bringing you these deep dives into cutting-edge science. Today, we're embarking on a truly captivating journey. It takes us right into the very core of our cells specifically, the mitochondria. Now, you might know them as the powerhouses of the cell, right? But what if they're also holding a kind of profound secret about how we age? New, groundbreaking research suggests these tiny organelles aren't just energy factories. They might be sophisticated, body-wide molecular clocks. We're going to unpack a remarkable study published recently that maps out this incredible phenomenon called mitochondrial clonal mosaicism. And he did this across an unprecedented 47 human tissues from hundreds of individuals. That's huge. Our mission for you today is to truly grasp how these tiny structures accumulate changes throughout your life.
Right. What those changes mean for different parts of your body, and what this deep dive reveals about the unique tissue-specific vulnerabilities that emerge as we all grow older. Okay, let's unpack this then. Before we get into this fascinating clock idea, let's quickly refresh our memory on mitochondria themselves. What are these integral structures, really? And why are they so central to cellular life and, as we'll see, potentially to aging?
Right. Well, mitochondria are indeed these ancient power generators residing within nearly all our eukaryotic cells. It's believed they originated maybe about 2 billion years ago from an alpha proteobacterium. You can think of it as a very early free-living bacterium. And this happened through a remarkable endosymbiotic event. Basically, one primitive cell engulfed another, and instead of digesting it, they formed this symbiotic partnership that, persisted through evolution. What's truly remarkable even today is that they retain their own distinct circular double-stranded genome. We call it mitochondrial DNA, or mtDNA, and it strikingly resembles bacterial DNA.
Huh. Still like his ancestor.
Pretty much, yeah. Now, this mtDNA is quite unique, and this is really crucial for our understandings of aging. It mutates at a rate anywhere from 100 to 1,000 times higher than our main nuclear DNA, the DNA in the cell nucleus.
Wow, 100 to 1,000 times. Why so high?
Well, primarily due to a lack of histone protection. Nuclear DNA is carefully wrapped around these proteins called histones, like thread on spools, which protects it. mtDNA doesn't really have that shield. Plus, it has very rapid replication cycles and less efficient DNA damage repair mechanisms.
Okay.
Think of it like this. Your main DNA is stored in a very secure, well-maintained library with lots of librarians checking for errors. But mitochondrial DNA, it's more like a constantly updated, high-speed online database with fewer checks, so typos accumulate much faster.
Right. Mistakes creep in easily.
Exactly. And this inherent sensitivity makes them an incredibly precise aging clock. They meticulously record this detailed, tissue-specific chronicle of damage accumulated over a lifetime. It's like a cellular diary of wear and tear, unique to each part of your body.
So, okay, what does this mean for us practically? Each cell has like hundreds or thousands of these mtDNA copies.
That's right.
And they show huge diversity, heteroplasmy, you called it, within cells and across the body.
Yes, precisely. Heteroplasmy.
So given that, how does this diversity and specifically the buildup of mutations change as we get older?
Well, this study reveals a highly dynamic process. You have these mutant mitochondrial clones constantly arising, and their abundance, it shifts significantly with cell division and also with mitochondrial turnover. That involves processes like mitophagy, basically recycling damaged mitochondria fission, where they split, and fusion, where they merge.
Okay, constant change.
Constant change. And these shifts are driven by two main mechanisms. First, replication errors during cell division in tissues that constantly renew themselves, like your gut lining. And second, mitochondrial turnover, which includes a process called relaxed replication.
Relaxed replication, what's that?
It's a unique and important process where mtDNA can actually replicate independently of the main cell cycle. So even in cells that don't typically divide, like neurons, their mitochondrial DNA is still being copied and potentially accumulating errors.
Got it. Even if the cell isn't dividing, the mitochondria inside are still active.
Exactly. And the expansion of these defective mitochondrial clones, it's directly linked to a range of diseases, particularly in tissues with high energy demands. We're talking conditions like Alzheimer's, Parkinson's, macular degeneration, diabetes, heart disease.
So this patchwork of mtDNA, the mosaicism,
right,
it's not just a marker of aging, it could be driving some of these diseases.
It certainly points that way. It seems to be both a driver and a powerful indicator of these underlying forces of aging.
Okay, here's where you said it gets really intriguing. The study's core finding. This clock isn't uniform, it's biphasic, meaning it operates in two distinct ways depending on the tissue type. Tell us about these two phases.
That's right, biphasic. So in the first phase, we're looking at what we call proliferative tissues. These are tissues with constant cellular turnover, always renewing themselves. Think about the lining of your gastrointestinal tract or your skin. These tissues show an accelerated accumulation of sporadic mutations and their clonal expansions.
Sporadic meaning random.
Random, yes. They occur randomly throughout the mitochondrial genome, and they're primarily driven by errors during that rapid DNA copying process. Specifically, they're often C to T and T to C transition simple base swaps during replication. So as these cells divide over and over, these copying errors just build up and expand within clones, creating patches of cells with these mutations.
Okay, so random errors pile up in renewing tissues. Can you give us some specific examples from the study? Which tissues are most affected, and what does that mean for our health?
Absolutely. The liver, for instance, showed the most rapid accumulation of these sporadic mutations. We're talking an average of 0.3 new somatic mutations acquired during life, not inherited per year. This can lead to an average mutant heteroplasmy level of like 15% by age 60.
15%. That sounds like a lot for the liver.
It is significant. And the esophageal mucosa, the inner lining of your esophagus, also shows a much higher mutation rate compared to the esophageal muscularis, the smooth muscle layer right next to it. This really highlights how strongly constant cell turnover drives this kind of mutation accumulation.
And the implication?
Well, this mechanism seems to make these rapidly renewing tissues more susceptible to age-related problems like tumorigenesis, tumor development, and just general dysfunction. What's more, these specific sporadic mutations are significantly enriched for potentially deleterious effects. They seem more likely to cause harm. They pile up specifically in these epithelial tissues, liver, esophageal lining, adrenal glands, suggesting a higher burden of potentially damaging mutations in these areas.
Okay, so that's one side of the clock.
Yeah.
Rapid renewal, random errors, potentially harmful consequences. What about the other side? The parts of our body that don't constantly divide.
That's the second phase, and it's quite different, yes. In contrast, we have post-mitotic tissues. These are cells that have largely stopped dividing. Think heart muscle cells, brain neurons, skeletal muscle. These tissues accumulate mutations too, but at specific predictable sites called deterministic hotspots.
Deterministic hotspots, not random anymore.
Not random, no. They're recurrently mutated sites, almost like predetermined weak spots in the mtDNA sequence.
Why there? What causes mutations at these specific spots?
It seems to be a consequence of the cumulative burden of high energy demand in these tissues, combined with rapid mitochondrial turnover, remember that relaxed replication? That, coupled with inherent error tendencies of the DNA copying enzyme, DNA polymerase gamma, during that relaxed replication process. So basically, intense energy use, constant mitochondrial renewal, and specific quirks in the copying machinery lead to these targeted mutations building up over time.
Okay, that makes sense. But this raises a big question we often hear about aging oxidative damage. Reactive oxygen species, ROS, the whole wear and tear idea. Doesn't that drive mutations in these high-energy tissues like the brain and heart?
That's a great question, and it leads to a really fascinating, maybe counterintuitive finding from this study. While ROS-related damage, oxidative damage, is prevalent in these energy-intensive tissues, and it does occur at some hotspots, it surprisingly does not significantly accumulate with age in these specific tissues, according to this data.
Really? So the body's cleaning it up effectively?
It strongly suggests that yes, that the body has remarkably robust repair or removal mechanisms for this specific type of damage in these tissues. It kind of pushes back against some older ideas. Also, another notable point, mutations in these high energy demand tissues tend to cluster in the noncoding D-loop region of mtDNA.
The D-loop, that doesn't code for proteins, right?
Exactly, which suggests a potentially less damaging functional impact compared to those sporadic mutations in renewing tissues, which often hit protein coding genes. It hints at maybe a unique resilience in how these vital non-dividing cells manage their mtDNA over the long haul.
Wow. So it's incredibly nuanced. Aging and tissue type are huge factors.
Yeah.
But the study also looked beyond our internal biology, didn't it? What about external factors like environment or even biological sex? How do they influence these patterns?
They definitely play a part, yeah. It adds another layer to this personalized aging picture. For instance, take sun-exposed skin. They found that individuals with non-L haplogroups,
Okay, remind us what haplogroups are.
they're broad groups based on mitochondrial DNA lineage. Non-L haplogroups are typically associated with lighter skin pigmentation, more common in, say, European and East Asian populations. These individuals showed a substantially higher mtDNA mutation burden in sun-exposed skin compared to those with the L haplogroup, which is primarily found in sub-Saharan African populations.
So lighter skin, more UV damage leads to more mitochondrial mutations there.
That's the very likely explanation, yes. It really underscores the huge impact of environmental factors like UV radiation on our cellular landscape, literally shaping how our skin ages at the mitochondrial level.
And what about sex differences?
Also quite distinct patterns. Females, for example, show a substantially higher mutation burden in breast tissue compared to males, and this difference seems to be established quite early in life, interestingly. And you see different things in reproductive organs, too. There's significant age-dependent accumulation of mutations in the testes, but notably not really in the ovary.
That's fascinating.
It really emphasizes why considering biological sex is crucial when studying tissue-specific aging. It's definitely not a one-size-fits-all model across the sexes either.
This deep dive has really painted a picture of just how dynamic, complex, and, well, personalized mitochondrial mutations are across our bodies. What's the big takeaway for us, for the listeners? What does this incredible research mean for how we think about aging and health?
Well, I think what's truly fascinating beyond just cataloging the mutations is the resilience of the mitochondrial machinery itself. Despite all these mutations piling up in different ways, the systems responsible for copying mtDNA, reading it transcription, and making proteins from it translation, they demonstrate remarkable tolerance to these genetic variations.
So the system can handle a fair bit of damage.
It seems so. For example, they found some predicted loss of function mutations, ones you'd think would completely break something, actually have alternative downstream sequences or workarounds that can compensate. It highlights this unique ability of mitochondria to tolerate certain changes and adapt. It's quite striking, especially compared to how sensitive our nuclear DNA systems can be.
So what's the bottom line for you and me? This idea of mitochondria as this two-phase clock, recording stress differently in every tissue. What does it unlock?
It gives us a whole new map, really, a map of tissue-specific vulnerabilities during aging. It's not just about getting older generally, but how each specific part of your body ages differently, with its own distinct clock ticking away based on its function and renewal rate.
Which could lead to better diagnostics, treatments.
Exactly. This deep dive could absolutely open doors for earlier diagnosis, maybe much more targeted interventions for age-related disorders. It moves us towards truly personalized approaches to healthy aging. You treat the specific vulnerability in the specific tissue. This study really highlights how digging into these intricate, sometimes surprising cellular dances can lead to potentially revolutionary insights into human health.
It's amazing stuff. Thank you for joining us on this incredible deep dive into the fascinating, complex world of mitochondrial aging. It's certainly a powerful reminder of the intricate biological clocks ticking away inside each of us. We really hope this deep dive has given you some profound aha moments and maybe a new perspective on aging. For more exciting research breakdowns and scientific explorations like this one, please remember to like this video and subscribe to the Sand Science Experience. Your support really does help the Sand Science Experience continue to explore the most exciting new research papers and preprints just for you. Until next time, keep exploring and stay curious with the Sand Science Experience.